So welcome to this May session of the Young Seminar series of the Italian Italian Society of Statistical Physics. Today, as usual, we have two speakers, and we will Marco Giulini and Arsham Gavassier. And we will start with Marco Giulini. And then after his talk, we will have the talk of Arsham Gavassier. So let me say a few words about Marco. Marco is currently a postdoctoral researcher at Utrecht University in the computational structural biology group of that university. He got his PhD in physics last year in January 2022 at Trento University. And he also had a master's degree in quantitative and computational biology, but his bachelor is in physics, if I understand. So, and he will give us a talk on from high to low resolution, the mapping problem in coarse-grained modeling. So please, the floor is yours.
Thank you for the introduction. Yeah, let me start sharing. Can you see my screen now?
Yes, yes.
Can you see the slides?
Yes.
Can you see the slides now? Okay, perfect.
Yes.
Okay, thank you for the introduction and for the invitation. I'm very happy to give this seminar. I'm a postdoc since 1 year, and what I'm doing now in the Netherlands cannot really be fit into the field of statistical physics. So I will tell you something about my PhD project, which was around the process of dimensionality reduction in coarse-grained modeling of biomolecules. And let me start with a brief outline of my presentation. I will start with a very broad introduction to the field of atomistic and coarse-grained modeling, and then I will discuss a bit the concept of information loss in the theory of coarse-graining. And then I will move to the focus of my PhD, which is this quantity called mapping entropy. And then I will give some conclusions and personal future perspectives. So probably not many of you in the audience know it, but it's pretty exciting to be a computational biophysicist now. This is because mainly because of two reasons. The number of experiments that have been carried out in the last years led to the deposition of a huge number of experimental structures in publicly available databases such as the PDB. And on the other hand, this huge amount of experimental data gave rise to the development of extremely accurate structure prediction algorithms. For example, Google's AlphaFold. Sorry, there is a lot of noise in the background. Can someone turn off the microphone, please? Yes, thank you. No, yeah. Yeah, better. Google's AlphaFold 2 is the main one of these super accurate algorithms. RosettaFold is another one. They really were a revolution in the field of computational structural biology. Basically, how do they work? You take the sequence of a protein, and after a lot of complicated manipulation, these algorithms give you a model, three-dimensional model of the protein. But of course, this is not the end of the story, as biomolecules and proteins in particular are intrinsically dynamical objects. And typically, the way we study the dynamics of such objects is by means of atomistic molecular dynamics, which is based on the numerical solution of Newton's equation of motion of the bimolecular system, and the Hamiltonian governing this time evolution is typically derived from a combination of experimental data and ab initio quantum mechanical calculations. And the applications of atomistic molecular dynamics are just ubiquitous. You can study, for example, protein folding with it. You start with the unfolded sequence of amino acids over the protein, and you hope to recover the three-dimensional structure. You can use MD for analyzing the different conformations of a protein. So imagine a protein has a closed conformation and you want to sample the open conformation. You run some molecular dynamics and you observe the results. And applications in drug discovery as well are really important and were actually crucial for the fight against the SARS-CoV pandemic. And there have been notable advancements in the field of atomistic molecular dynamics in the last years, mainly the GPU implementation of the most popular molecular dynamics softwares, and the distribution of molecular dynamic calculation over a grid of users, so distributed computing-based molecular dynamics, and the development, the construction of a special purpose architecture such as Anton tree for running molecular dynamics calculation. Of course, this algorithm has some drawbacks. For example, the fact that biologically relevant time and length scales are still very hard to reach with plain all-atom and molecular dynamics. Second, the fact that this algorithm, that a molecular dynamics simulation gives rise to terabytes, gigabytes, and terabytes of data, which are hard to store and to analyze. And the third, all-atom MD is not really a democratic method, let's say, as in order to perform meaningful simulations, research group has to have access to a high-performance computing facility, and which is not to be given for granted. And if you can run longer simulations, you get typically better results, and this creates a sort of an inequality. And one way to get rid of these drawbacks is to develop coarse-grained models. What is a coarse-grained model? Basically, it's quite simple. You start from an atomistic, fully atomistic model of your system, which is your ground truth, and then you apply a mapping operator to this system. So you reduce the dimensionality of the system, and then you apply the coarse-grained force field, the coarse-grained effective interactions to the degrees of freedom that survived this mapping procedure, and then you're left with a coarse-grained model. Now let me focus on a special category of bottom-up coarse-grained models, which are bottom-up coarse-grained models. Basically, the idea of such models is to exploit information coming from a detailed, fully atomistic model to build the coarse-grained model. How does this work? This is pretty tricky, but I hope to be able to explain this to you. Basically, we start from the fully atomistic system, and in the fully atomistic system, we know that we can use the Boltzmann probability distribution to describe the probabilistic weight of each configuration. And then from this Boltzmann probability, we can calculate the probability of a coarse-grained configuration, in this case, big R, simply by integrating the atomistic probabilities P of small r over all the configurations that map onto big R, as it is shown in this picture. And an important quantity in this context is what is called the many-body potential of mean force, which is basically found by taking the logarithm of such coarse-grained probability. This is indeed the target of such bottom-up coarse-grained modeling. So this is the quantity that we want to find while doing this specific coarse-grained modeling. And why? Because this U0, this many-body potential of mean force, samples the coarse-grained probability distribution P of big R that would be sampled by the reference system by our ground truth once observed through the glasses of the coarse-grained mapping. There is enough literature that proves that this quantity, the many-body potential of mean force, is impossible to calculate. I'm not even talking about simulating for complex systems, and typically one has to resort to an approximation U of this potential, which is a sum of a few body components. And so this is the scenario in which we are typically, while developing a bottom-up coarse-grained model. You have your exact coarse-grained model, so the one that corresponds to the many-body potential of mean force, U0, and an approximate coarse-grained model in which the interactions are parameterized using U. And of course, we need a measure of distance that quantifies how these two models relate. And this is essentially a measure of the goodness of the coarse-grained interactions. How well is the potential U, how close is U to U0? And there are different ways to determine this distance. For example, the inverse Monte Carlo method, the iterative Boltzmann inversion, and more recently, the multi-scale coarse-graining method and the relative entropy approach. I will discuss now a bit this last approach, the relative entropy one. In my opinion, it's super elegant and super general. Basically, the relative entropy is a Kullback-Leibler divergence between the probability distributions generated by the two models. So the reference probability is, of course, the one generated by the many-body potential of mean force, while the probability that is compared to this one is the one generated by the approximate coarse-grained model with the potential U. And this is nice, but of course, if we think about the overall aim of coarse-graining is to find a coarse-grained model which is as close as possible to our ground truth, to the atomistic model, to the fully detailed description of the reality that we have. So this is actually the side of the triangle that we aim at minimizing. And indeed, saying that the distance between the exact coarse-grained model and the approximate one is 0 does not imply that we reproduce the properties of the atomistic system. Indeed, we have to consider what lies on the other side of the triangle. And this is basically the distance between our first principle theory, the atomistic model, and the most accurate theory at low resolution. So this distance is basically a measure of the goodness of the coarse-grained mapping, of the choice of degrees of freedom that I employ to reduce the dimensionality of my system. And what we found out in this review a couple of years ago is that in all the existing coarse-graining procedures, the mapping is almost always given as an ingredient, as an input to the coarse-graining procedure, rather than an output, than something that has to be optimized and searched for. Therefore, if we employ the relative entropy as a distance between the two coarse-grained models, the exact and the approximate one, we can further decompose it in two different Kullback-Leibler divergences. In the first one, we see it's a Kullback-Leibler divergence between the probability distribution of the atomistic model and the one of the approximate coarse-grained model. So it contains both the mapping and the approximate coarse-grained interactions, while the second, which has a minus in front of it, is a Kullback-Leibler divergence between the atomistic probability distribution and the probability generated by the many-body potential of mean force. And we call these two terms total S and SMAP, mapping entropy, and we can finally map them to the three sides of our triangle in the space of models. So now our aim is to inspect a bit the mapping entropy to understand this complex mathematical object. And it is possible to prove that we can further divide it in two terms, sort of a geometric term, which is basically almost equal to 0, and probability smearing term, which is important for our purposes. And what is the probability smearing term? Well, in this case, we are comparing in this equation of the mapping entropy the Boltzmann probability, so the probability of the atomistic model, to a smeared probability. What is this? Imagine that you are an observer lying on the low resolution configurational space on configuration big R in the top right. And here you know that some high resolution configurations, so some configurations in the atomistic model map onto you. And then you're asked to estimate the probability weight of such configurations in the high resolution configurational space. Of course, since you're sitting in the low resolution configurational space, you don't have access to the high resolution configurational probabilities. Therefore, the most reasonable assumption you can make about this high-resolution configurational space is that the probabilities of all the configurations are all the same and equal to your probability, the probability of the low-resolution configurations, just divided by the number of configurations that map onto it. And this is what the mapping entropy is measuring, the difference between the real atomistic probability and this smear probability given by this averaging. Now, the pictorial representation of what I just said. So we start from the original high-resolution representation of a system on the bottom left of the slide. Basically, in this case, the fully detailed picture of Monte Bondone in Trento. And then we apply some filtering. In this case, the mapping process does the filtering. And then we reconstruct a high-resolution representation. But of course, we need to average in the points that were removed by the mapping. And the mapping entropy quantifies indeed the impact that the choice operated by the mapping had in terms of reconstructing the original high-resolution representation. In this paper, a couple of years ago, we proved that this complex object can be optimized, can be calculated in a much simpler way without making use of super high-dimensional probability distributions such as PR and P bar of R. And in this case, we proved that it's possible to approximate this quantity simply by an integral over the Cosgrane state of the variance of the atomistic energies belonging to that state. And then we said, okay, we have a way to calculate this quantity, and now we can think of minimizing the mapping entropy. And so we developed a protocol for this minimization. We start from what is called a random mapping, in which we have high value of the mapping entropy. And then we feed this random mapping into a simulated annealing minimization procedure in which the mapping is changed and the mapping entropy is measured and the moves are accepted or neglected according to our Metropolis criterion. And then in output, we have an optimized mapping characterized by typically a low value of mapping entropy. And now we can basically inspect these optimal mappings, the different solutions that come out of this optimization procedure. And of course, we are interested in those atoms that were never conserved by the optimization, and of course, in those atoms that were always conserved, which are their properties. So let me start with the first actually optimization that was performed according to this protocol. We performed a mapping entropy minimization over this tau-mapping protein. This is the toxin of the Indian red scorpion, so quite small peptide, like 31 amino acids, sort of like 500 atoms in total, and we wanted to reduce the dimensionality of this peptide. What we found out is that the atoms that were always conserved by the mapping entropy minimization procedures are actually the atoms that are involved in the biological function of this protein. Indeed, these two arginine residues were pinpointed as extremely relevant from the mapping. So you shouldn't really never remove these things, And basically, these are the two main amino acids that are involved in the binding to the substrate of this toxin. And this was rather surprising. And of course, we wanted to prove this the generality of such approach, and we moved to a bigger system, the adenylyl kinase enzyme. This is quite important protein in our body, as it performs the interconversion between the ADP and ATP in the cell, and also in this case, the completely unsupervised minimization of the mapping entropy said that we should never remove atoms belonging to arginine 88, arginine 36, and lysine 57, which were all related to the catalytic activity of the enzyme. Basically, if you mutate these two arginine residues, you never you won't get the correct interconversion of ADP into ATP. And also lysine 57 is very important in blocking the release of the adenine molecule from the hydrophobic pocket of the enzyme. And another success story of this protocol is given by the alpha-1 antitrypsin protein. Also in this case, this is a much bigger protein. It has like 6,000 atoms, more or less. And also in this case, upon mapping entropy minimization, we got two residues that were pinpointed as really, you should never remove those. And also these two residues are important for the activity of the protein. A mutation on arginine 101 can lead to severe deficiency in alpha-1 antitrypsin, and basically methionine 358 is basically the reactive site of the protein. If you oxidize it, if you modify this residue, you can have pulmonary emphysema. This is what happens to heavy smokers, for example. Why is this happening? Well, we hypothesize that all these conserved amino acids have quite high conformational entropy and are subject to strong energetic fluctuations, so that upon the binding of a substrate, both this conformational entropy and these strong energetic fluctuations are sort of suppressed. That's why they are particularly favorable for the binding. And it's important to underline that all the data used for this minimization protocol, for the mapping entropy minimization, come from simulations that are performed in absence of the substrate, in absence of the partner of these proteins. So we can use this tool as an unsupervised analysis of like trajectories, substrate-free trajectories, to identify potential hotspots of a protein, of unknown proteins. Then the last, probably the last thing before concluding, which is the idea of applying this quantity, the mapping entropy, to the analysis of complex systems, since it is working so well in the realm of coarse graining, which is a very complicated subject. Maybe we can take this variable and use it for the analysis and feature selection of the time evolution of complex systems. In this case, we do not really need the approximation of the mapping entropy. We can take its original formulation in the Kullback-Leibler sense. We just want to consider the Kullback-Leibler divergence between the fully detailed distribution, the fully detailed probability distribution of the full system, and comparing to the smeared probability. And in this paper, in PRE last year, we applied this quantity to two different systems, a system of non-interacting spins, and an approximate model of the Nasdaq financial market. And also in this case, we discovered that mapping entropy gives us information about the subset of degrees of freedom that we should employ to observe the system at low resolution. And this, of course, is another proof that this quantity is extremely powerful in the completely unsupervised analysis of some data about complex systems. And yeah, so this leads me to my conclusion. Here on the top right, you can find a link to the GitHub code GitHub repo with all the code performed to run all these calculations. And well, I will start by saying that this quantity, the mapping entropy, quantifies the distance between first principle theory, in our case, the fully atomistic model of a biomolecule, and the most accurate effective theory at low resolution, in this case, the many-body potential of mean force. We understood that we must employ optimized mappings, optimized descriptions in order to construct accurate coarse grain models. And taking the mapping entropy outside of the realm of biomolecular coarse graining, we can use it to perform an optimal coarse graining of arbitrarily complex systems. And with this, I would like to conclude and to acknowledge my former colleagues in Trento, without whom this would have never been possible, and also my international collaborators at University of Santa Barbara, University of Pisa, and the Weizmann Institute of Science. Thank you.
Thank you. Thank you very much for the talk. Are there comments or questions? I have one comment and one question. One comment is about, indeed, we used a few years, well, probably several years ago, also the Kullback library measure just to quantify the information present in a correlation function in a system. And in your last slide, one of the last slides, you were saying,
I just got the sharing. Yeah.
Okay. You were citing the application of similar approach to complex system, and in particular to financial markets. Indeed, the application we did was also to a correlation matrix of a portfolio of stocks. My question is, when you say when you did your application, did you assume some type of distribution for the P of R, or this is just something that it is left as provided by the data?
Yeah, we leave it as provided by the data. Like the observations that you collect are basically defining your P of R, the probability in the high resolution system, let's say. So yeah, if the data is bad, if the data is not at the equilibrium as the stock market typically is, of course, there is a few chances of getting something meaningful out of it. In our case, we were able to get a meaningful dimensionality reduction, but which was
dimensionality reduction of what in the case of financial market?
In that case,
what was the quantity that you analyzed? Return or price?
No, just the mapping entropy. Like the mapping entropy is nice because it's unsupervised. Like you take the
no, I understand, but the mapping entropy of what? What was the system?
A combination of you take the full market as a reference. That's your high resolution system, and then you take a combination of the stocks that make up this market as the mapping, and this induces a low resolution representation, and you want to optimize the low resolution representation so as to reproduce the properties of the high resolution system as the full stock market, let's say.
So in a sense, you created some indices and you wanted to be as representative of the stock as much as you can.
Yeah,
I understand.
Yeah, typically, like what does the FTSE MIB, for example? Like you take a subset of stocks and you want to be representative of the full market. Yes.
Other questions or comments? Okay, if not, I think we thank again Marco for this presentation. Thank you very much.
Thank you, and thank you for the invitation.